Hi, my name is Brittany Jasperse, and I'm a postdoc in the lab of Helen Lazier in the Department of Microbiology and Immunology at UNC Chapel Hill. I'll be sharing a little bit about my work on host genetic determinants of Powassan virus pathogenesis. Vector-borne diseases are spread by the bites of infected arthropods, such as mosquitoes and ticks. Common vector-borne diseases include West Nile virus, a flavivirus and the most common cause of mosquito-borne disease in the continental U.S., and Lyme disease, which is transmitted by the bites of ticks infected with the bacteria Borrelia burgdorferi. An emerging vector-borne pathogen in the U.S. is Powassan virus, a neuroinvasive flavivirus closely related to West Nile virus, but that is transmitted by the same ticks that carry Lyme disease. While cases of Powassan infection are still rare, its incidence is increasing. Similar to West Nile virus and other neuroinvasive flaviviruses, Powassan infection leads to a broad range of outcomes in humans. Approximately 80% of infections are asymptomatic, and about 20% lead to an acute febrile illness. A subset of symptomatic infections progress to neuroinvasive disease, including encephalitis, meningitis, paralysis, and death. Approximately 10% of reported encephalitic cases of Powassan infection are fatal, and of those that survive, about 50% experience long-term cognitive and functional sequelae. Importantly, the factors that influence susceptibility to severe Powassan disease remain unknown, but one potential explanation is the genetic differences between infected individuals. To study Powassan virus in the lab, we use mice as a model organism. Traditionally, researchers use strains of genetically identical mice, which would be like studying Powassan disease in identical twins. But in our case, we're trying to capture the genetic diversity seen in human populations, so we instead use the collaborative cross. The collaborative cross is a mouse genetic reference population, which was generated by crossing 8 different strains of mice, including 3 wild-derived and 5 classical laboratory mouse strains. These founder strains were bred to generate roughly 100 reproducible mouse lines that have genetic contributions from each of the founder strains. Because of this, the collaborative cross panel of mice reproduces the genetic diversity and phenotypic range seen in human populations. To identify host factors that influence susceptibility to Powassan virus infection, we infected a panel of collaborative cross mouse lines and observed a range of susceptibility to Powassan virus, including 100% lethality in 11 of 17 lines, 5 lines with intermediate lethality, and 1 resistant line. We narrowed our focus onto 2 of the mouse lines, 1 susceptible line and 1 resistant line, and now we can ask the question, which of the genetic differences between these 2 mouse lines are responsible for the differences in susceptibility to Powassan virus? To narrow down which specific genes that are different between these 2 mouse lines are actually responsible for the differential susceptibility to Powassan virus, we can breed these mice together and generate F2 progeny. The reason that we study the F2 generation is that when the collaborative cross lines were generated, they were bred to homozygosity. So the F1 mice will all get 1 copy of each chromosome from each parent, but the F2 generation is where we'll start to see variation, and each F2 mouse will have a unique mix of the genomes of the F0 lines. We can see this visually by looking at a phenotype like coat color. So, for example, we bred a white mouse with a black mouse, and we get F1 mice that are all black. And then you can cross those F1 mice together, and now in the F2 generation, you have black mice, white mice, and we even see the emergence of new phenotypes like brown mice. So going back to our Powassan studies, these F0 lines represent a line we know to be susceptible or resistant to Powassan infection. So we generated our F1 and our F2 mice, and we infected F1 mice with Powassan virus to answer the question, is resistance to Powassan infection dominantly inherited? And the answer is yes. And then we infected several hundred F2 mice with Powassan virus to assess susceptibility to Powassan virus infection and determine the distribution of phenotypes, for example, time to death, which gives us an idea if there's a single gene or multiple genes responsible for the phenotype. And we're at the stage now where we will genotype the F2 mice and use quantitative genetics approaches to identify genes that are associated with resistance to Powassan infection. Our goal is to further the understanding of flavivirus neuroinvasive disease by identifying host genetic factors that modulate Powassan susceptibility in mice and provide insight into factors that impact neuroinvasive flaviviruses more broadly. Thank you to my lab members, especially Melissa Maddox and Michaela Dramini, and to our funding sources.